Sorry. Oh, hold on. I don't know if, hold on.
I'll record too, if that's cool.
Can you guys record? Wait, hold on.
Yeah, it says I need to get permission from host.
Yeah.
Okay, hold on.
Thanks.
Okay, who wants to record? Just put your hands up. Let's do this like class. Who wants to record? There you go. Okay. Think. Okay. Let's see who else. Is everyone good? No one else wants to record? Just asking. Okay. And time. We're good? Okay. So we're gonna go over DNA sequencing, and we're gonna go over how to find your amino acid and the CRP protein and any interacting amino acids. Okay, so for PC, you're gonna have to download.
I was able to get Chimera to work, remember?
Yeah, for DNA sequencing, you guys need chromas for PC users.
Okay.
Mm-hmm.
I do have a question though. Have you guys tried the POV Ray? Do you guys know if that one is Apple friendly?
That one isn't Apple friendly. The only one that we're able to use is Chimera.
Because you know how he says is either the WISP PBD viewer or Chimera, and then he said that for us to do an image, to do the figure of the molecule images, we need another program. I think it's POV Ray. So I just don't know if that one is friendly or not. If it's not, I can ask the professor.
I think for the POV Ray, that one is not Apple friendly like Mac at all. I think we had discussed that in class when we were like barely starting to do DNA sequencing, and he said that it was fine if we just use Chimera.
That's the one to make the image like more like high resolution, right?
Yeah.
Okay. Yeah, he said it's not like if you have a Mac, because I have a Mac, he said it wouldn't work for Mac. But he said like for my, because I asked him for my paper, like if we have to have like those high resolution pictures, and he said you can just screenshot it, like it doesn't have to be through that.
Okay, cool. Thanks.
Mm-hmm.
Okay, so we're good. Anyone joining in, do you guys want to record or are you guys fine? Okay. So
I would record.
Okay. Hold on then. Let me just give you permission to record. There you go. You should be able to record now. All right. So we're going to start. So I don't know if you guys want to have a Word doc open already so you can like start putting all your information. But on Canvas, we all have our DNA sequence, which would be this. This is for 4 peaks for Mac. I don't know. Sorry, I don't know how the Chromas one looks, but this is for the Mac one. You guys all have this file?
Yeah, and for PC, it basically looks the same. So,
Okay.
Yeah.
So from there, we're going to.
And then which one is this one under in the Canvas?
This one is under modules.
Yeah, be DNA sequencing.
Mm-hmm.
Okay, thank you.
Welcome. And so from there, we're going to copy our sequence. For that, you can go to, for 4 peaks, you can go where it says edit and then it says copy sequence.
It's the same on PC.
Okay, thank you. Okay. And then,
And do we want that in FASTA
yes.
or FASTA? Okay.
Yeah. And so can you guys see this now?
Yes.
Okay. And so what you're gonna do is that you're gonna place this in your Word docs. Okay, so just tell me when you guys are ready to go to the next step. Okay, so everyone's should be good. Do you guys need a couple more minutes? Oh,
So for this one, we're using Four Peaks, right? Which is the one we had already. Well,
yeah, the one
Yeah.
you should have. Mhm.
Mine, I did it in Chromis. That's okay, right?
Yeah, that should be fine.
Okay.
Okay, and then from here, we're gonna find our start and stop codon. So if you guys are on the Word doc, you guys can click on the little search button here, and then it's ATG. So that first ATG right here, that one is your start. And so you can delete all that there. And then your stop should be, hold on. Sorry guys, I totally forgot the stop codon sequence.
It'd be like TAA.
No, it's a whole different one. Hold on, I have it somewhere.
it's right there where it says CATTAA.
I know, yeah.
To the left of that same line you were at.
Left of the same line.
Okay, you see where all the ends are?
CA.
Above that? To the right. Left?
To the right, right here, right?
Yeah, that area right there.
Sorry guys, yeah, that's our stop. So let's highlight that and then you can just delete all this extra. Oh, not the whole thing. Why is it doing that? Just this. There you go. Okay, so I don't know if any of you guys have ends within your DNA sequence.
I don't,
Have ends. Just making sure.
No.
If you guys have any ends, you guys have to go back to your Ford Preaks or chromas to see what it would be. Okay. And so this is gonna be our mutant. So we're gonna call it mutant.
Oh, I actually do have one random in, like almost before the end sequence.
Okay.
I just did a search for in and I had one.
Okay, do you want to share your chroma so we can see what nucleotide it is? Or do you feel like you know what nucleotide it is?
I'm not sure. Give me a sec here.
Okay.
Am I the only one that has an in?
I have ends too, but I already figured out which nucleotides they were.
Okay.
How do you find out what nucleotide it is?
You have to go on, well, because I'm a Mac, so mine is, what's the program for Mac?
Four peaks?
Four Peaks. There you go. You have to go on Four Peaks, and then wherever there's an N, like whatever position, you go on the Four Peaks and then look for the peaks, like the colors. I think it's like black, green, blue, and red. And then
Okay.
look for them like that. I forgot what the colors were.
Okay. I found the in.
I was just able to open up my DNA sequencing. How do I open it up on Chromas? I forgot. I just downloaded it right now because I got a new computer.
You should be able to, on Canvas, it should say download. And for PC, I don't know if it tells you like how to directly open it. Can you guys see this right now?
Can I share my screen? Because
Yeah.
I'm confused now.
Mhm. Go ahead.
Okay, can you guys see my Chromas?
Yeah. So you go to edit
Edit.
on top here,
Okay.
and then it should say copy sequence.
Edit. And then can you guys see what I'm doing? Copy sequence.
Copy
Oh yeah,
sequence.
that's right.
And then the
And then
FAFSA. And then you just insert that into a Word doc.
okay. So then I would go to Word doc.
Mm-hmm.
Okay, thanks.
Welcome.
Okay, I found the N and like the peak and stuff, and it's pink. I just don't know how to figure out which one that refers to.
Pink. We don't have pink. What colors do you have? Pink, blue, green, and
I have
black?
and green, red, black, blue.
Okay, so for that peak, what color do you think that is? It doesn't
It's
have
pink.
like
It's like fuchsia. It's pink. It's fuchsia.
fuchsia. Hmm.
Like a hot pink.
I think you would have to go with red then, right?
Yeah, I think so. I
Yeah.
think T.
T.
Yeah,
Mhm.
if you don't mind sharing, you could share. We can try to find what nucleotide it is.
Yeah.
Yeah, let me see how to do this here. Share.
That's interesting that it's fuchsia because even the ends that I have, it's still the same color, but it's put.
Okay, can you guys see that?
Yes, we can.
Oh,
Okay, so there's my N,
so
and it's fuchsia.
I think what they mean is the peak. What color is the peak? So if you put your cursor above the N, or how do you?
I think you'll have to like go down to where the peaks are. Like you can try to go all the way down if you follow your mouse.
I think that's a black right there.
Yeah.
It's black.
Is
Is that it right there? I'll try to draw a straight. Oh, here, I can do this.
Oh,
It's
That's a G.
black.
Mm-hmm. That's a G.
A G.
Mm-hmm.
So just write down that it's a G, or like, what does that mean?
Yeah, well, it's nucleotide G, but sometimes for some reason they mark it as N because they can't distinguish it, probably because it's a really low peak.
So then on your word, you would replace that N with a G
Mm-hmm.
because you
Okay.
you read that yourself instead of it manually doing it.
Okay, so let me share my screen. Going to file. We're gonna go to the NCBI to get our CRP for our wild type.
Wait, I just put it on the Word doc. Which one? Sorry. ATG is the stop codon, right?
The start codon, yeah. The first ATG is your start codon.
And the stop codon is
The CATTAA.
okay.
So I have a question. So the ends, if the peak is black and it's aligned, then we assume that it's a G and we just switch that?
Yeah, for any of the ends that are within your DNA sequence from start to stop, if you have an N, you look at the chromas or 4 peaks and then try to see where that N lands on. And then whatever color that peak is, it's that nucleotide.
Okay.
So what
Yeah,
if,
I had an N and it was black peak, so that was a G.
okay, so what if it doesn't land in any peak though?
It doesn't
Like-
land in anything.
No, it's like between peaks.
Can you share so we can see?
Yeah.
If you click and hold your mouse down, a line should appear, and you
I
can
gotta-
kind of line up.
Okay, let's see. No, it falls right in between. Like,
Oh, snap.
Yeah, can you
well,
share
there's
it?
one of them. Okay, this one is blue, but the other one falls right in between. It's like nothing between it.
You want
Okay,
to try to share so we can try to
Yeah.
help you? It's like a little green button at the bottom of the share screen. Yeah.
This one right here. Can you see it?
And yes,
See, it's like right between the black and green.
But is that within your DNA sequence?
Well, that's big.
I had the same issue. You have to delete the stuff after the stop codon, and that'll get rid of those ends. You might have another end to the left more.
Mm-hmm. Because mine is like that too. Before I get to the ATG, I have a couple of ends.
Yeah. Did you find your start and your stop codons and delete the stuff outside of that?
No, I haven't.
Okay. Did you paste it in the Word doc?
I did. Oh, you
Okay.
can't see
So
it.
at the very beginning, you should find an ATG.
ATG.
Mm-hmm.
Yeah. Delete everything to the left of ATG.
Hold on. ATG. Okay. And from the left, I delete it, right?
Yeah. Everything to the left of ATG, delete that.
Okay.
And then towards the end, you should have a bunch of ends, right? Like a pile of them.
Yes.
Okay, so before that, look for a CATTAA.
CATTA. Okay.
And another A.
Oh, two.
CATTAA.
Okay.
And then delete everything to the right of that. That's your stop codon.
Okay. Got it.
Do you still show it in between the sequence?
No, I took it off actually. Okay.
Okay.
No, yeah. Okay. Let me stop sharing this. Mm,
So obviously you just did that on the Word document. It's not going to show in your
Yes.
Chromis or
My
4Peak. So just, you'll have to now search your Word document. See, do you have any ends now within those codons? I had one that was still within it.
No. That's it.
Okay, then you should be good.
All right. Thank you.
Okay. I think everyone's good with the start and stop codon now with their DNA sequence. Now we're gonna go to NCBI so we can get our CRP. So we're going to search up MG1655 and coli and CRP.
All of us search for that same thing.
Oh.
Yeah, all of us searching up the same thing.
Do you have to put gene on? Does it have to be specific for gene?
No,
No.
just
Okay.
be, yeah.
Sorry, it's just I have it on here like I've clicked it before, so I wasn't sure if that's what we used before.
So
When you search it, it'll give you the option. It'll show you genes versus proteins and all that.
Okay, thank you.
hold on. Are we good? Next step.
Yep.
So we search. We go to gene. CRP gene. We click on that. And then we're
Can you go back one page so I can see where you clicked?
TRB right here?
Thank you.
The ID?
DNA bind, oh, CRP with DNA. Okay, cool.
Then we click on that. And then from here, you can click, not click on it, but you can like point your mouse to hover on it.
Hover. Okay.
Mhm. and then you go to the FASTA record right here. And
Okay.
it's gonna send you to a whole different thing.
Can you show
then-
that one more
Yes,
time?
ma'am.
Sorry.
So you can go to CRP, and then you just need to hover it. You don't need to click it. You can hover it, and then it says FASTA record right here, and then you click on that.
Okay.
So after fast record, mine shows like a little bit different than yours. Does that matter? Oh yeah, it definitely matters. It's different.
So we all have the same one?
Yeah, because this is our wild type. So this is
Okay.
gonna be the same no matter what.
Oh,
The only
okay.
thing that's different would be our mutant. That's
Okay.
the one that we got from the 4 peaks or the chromas.
Got it. All right.
Hey, Merlin.
Yeah.
I can't find the one you clicked on.
I couldn't either. You can also click on top. There's something that says FAFSA. When there's that red little line that she was showing us to hover over.
No, I
On
mean the one
top.
before that, like the sequence you clicked on.
Did
Yeah,
you
that page.
all
Yeah.
this?
Yeah, MG1655 and coli and CRP, right?
Yeah, and then it should be the first one right here. It says CRP G90, and then it's a DNA binding transcriptional dual
Just
regular.
paste a link here. I'll just put a link for it in chat.
Okay.
There you go, Ty.
Okay.
Thank you.
That should take you straight there, hopefully.
Okay.
Thank you.
Yep.
That's weird, even though we're all doing the same steps. Not showing.
I know, it's like the same exact web page. It's weird.
And then you just go to FAFSA record. Oh, I did not see that there. Sorry, the participant thing blocked me. But yeah, you can either click that or you can like do it how I did it, which is hover your mouse over the CRP gene and then just click on the record and it should send you to here. And then from here, we're gonna copy all of this. Okay, and then just tell me when you guys are ready and you guys got the sequence for the wild type.
Okay, I'm good.
Everyone good? Okay. So we're gonna go back to our
So
Word
we
doc.
just copy that to Word. That's it, right?
And then you just paste it in there.
Okay.
Hold on. Let me fix. I don't know why it's like that. You know what, we're just gonna keep it in the original format. Sorry about that. Okay, and then from here, we're gonna align it. So I would try to make sure you guys are writing what you guys are doing, because I know for the report, we're gonna have to do this to identify mutant.
It's just a multiple sequence alignment, right, between the mutant and the wild type.
Yeah. So we're going to copy the mutant. Oh, wait, hold on. Copy the wild type first. And go to tea coffee.
Just DNA.
It's this one, the one in the middle that said simple.
Oh, I didn't know you could even click that.
Oh, sorry. Yeah.
And you would put the mutant, right? Or
You put your wild type and
Wait,
then
what?
your mutant. When you put them in the tea coffee, make sure you have the little symbol and then your mutant or like wild type to make sure. Then copy your mutant.
You just put a space between them in the box there.
Yeah, hold on, let's see if I can. Wait, I don't remember how to make sure it tells you what to do. This, yeah. And just copy your mutant and then paste that on the tea coffee.
Did you leave a space?
What's that, Lizette?
Yeah.
Did you guys leave a space?
Yeah, you want at least one line of space between them.
Okay. Is that
And-
weird? Does it show you when they try to talk?
Who, me?
Your box lights up yellow.
Yeah.
I was like, oh gosh.
Okay, and then is everyone ready to?
Does it have to have the little parentheses or like the little blue things?
It needs to have the greater than sign or it won't
Yeah.
recognize as a FASTA.
Okay, but anything other than that, like the little blue thing, like after mutant, does it need to have
No,
that?
that's
Oh,
just
okay.
to identify the space that I put.
Oh, okay. All right.
Yeah.
That's just her
No.
word format.
I believe that the professor, when he did it, I think he did the show. I think, what is it called? More options or advanced options on the tea coffee. And then he changed the residue number. He put it on, and the alignment length is 60. So show more option, yeah. And then he had the residue number on. Yeah, that's 60, and then the residue number is on.
Thank you.
Okay, give me a second. Is it more options?
Make sure you write that down like in our notes so we can type that in.
60. Okay.
Okay, and then submit. Oh. I think mine has extra space. Oh yeah, so that's what it was. Sorry. I don't know if you guys have that too.
No, mine's running.
Okay, as long as you guys are running.
So it was wild type and then mutant underneath it.
Fix that up again then. Oh, not that. I put wild type on top.
Okay.
Yeah.
Yeah, as long as they're labeled before the greater than sign, just so that you know which is which.
Okay. Let's see. Let me run that again.
And mine finished.
Yeah, mine too.
Okay, mine's done.
This is so helpful. You're now our adjunct professor. You're hired.
There we go. Now it's mine's running.
It only took like 30 seconds, not even that.
My Wi-Fi is like really slow. So it might take a little bit longer for me.
Did anyone take bioinformatics with FanLar?
No. I wanted to, but it was like 4 already.
I took it over winter in like 2 weeks. Oh my gosh, we had to do all this stuff, but it was crazy.
Oh, hell no.
So what you should see is this. You should have them red. If you have any other color, then something is probably up. But anything without an asterisk, so let's see. So we have one there, and then we have some right here.
My very last letter on row 586 is a yellow.
Like this?
Yeah, I mean, mine's a little different than yours, but yeah, just very right at the end, I've got a couple yellows, just like you do.
This should be fine because I remember he said that he added a bit more to it, a lot more histamine.
Yeah, it doesn't look abnormal. I was just checking.
Okay, so we're gonna copy this. Ooh, all of it.
The whole thing.
Whole thing.
I'm gonna screenshot that too. Seems like a good image to put in if he wants figures.
You know, mine came out really weird. You know how I said 60, but somehow it didn't come out 60. Mine came out 51, 102. So it came out like really odd numbers.
See, mine came out at 54. Hold on. Why does mine look like this? Oh, there you go. Still there.
What were the other options that we had to sit there and put before we submit our sequence?
What were the other options? We were supposed to hunt 60 for
the alignment link.
Alignment, yeah, and then the residue on.
Number on. Yeah.
Residue. Oh, okay.
Yeah, but for some reason it's not at 60. It's at 54 for mine. And I think someone said 58.
Where are you seeing the 60 versus 54?
Right here.
Oh, mine's 45.
Yeah, mine's 51.
Oh, snap.
I have 54.
Maybe 60 is the max.
Yeah,
it's
maybe.
Limiting it to 60.
I just realized on the when I had tried it to do it before while he was doing it, and it's not, the numbers aren't labeled. I don't know if that makes sense.
Yeah, so from what I remember from lecture, it was because we were on the wrong website. And so Michael and him were trying to figure out if that had to do with anything. But from what I remember, that doesn't really affect anything. So what I remember he told us to do was to just leave it. And then instead of that, you would go to more options, change it to order, or under the order, and then you put input. That's all. That's the note I have from the lecture that we did with when Michael was an example. So I don't know if that would change anything, but I didn't do mine as 60. I just left it to whatever it was.
And what was it that you said, Calissa, that he didn't?
So it wouldn't be advanced. You would just scroll down to where the advanced options were. You would click more options, and then it has 2 categories, and then there's one that says order. Yeah, so yeah, and then you would just put input. I guess it was already on for you, but it wasn't on for me, and then that's all I did. I don't know if it'll change anything for you guys, but even when I got my sequence, I didn't have numbers on the side.
Yeah. Yeah, it didn't make a difference. I think we should be good though.
Yeah, as long as you have your mutants or your changes, I think it should be fine.
Oh, wait. So I'm like looking at yours. So yours is all pink?
Yeah, it should be at least all pink. At the end, there could be a variation of color because he added a few more histamine.
Okay.
So, but overall, there should be reddish pink color.
Mine, I have a lot of yellow.
I think yellow, you should be good as long as it's not like green or blue.
It looks like yellow's average, so maybe you just have a very mutant.
Okay. Yeah, because most of it is all yellow. Yeah, because the pink it's like in the beginning and like a little bit like towards the end.
Can you share your screen so we can kind of
Yeah. Where do I? I gotta someone walk me through how to share screen. Oh, okay. Never mind. Got it. I'm like sitting there still trying to get used to this. So, can you see it?
Yeah. Whoa, yeah. Something's,
Yeah, see.
yeah, something's very, what?
So that's what I have.
Yeah, because you have stop codons within even your sequence. So you have
Show us your sequence, your two
there.
sequences.
Yeah.
Oh, can you show us your two sequences in your Word document?
Yeah.
So I have my mutant here and then I have my wild type.
And it let you go through with all of those empty spaces at the end.
Mm-hmm.
Wait,
Well,
which
maybe the issue is that they're not like aligned the same.
Yeah.
Yeah. So click the little where it says normal for the Word doc. You see that little space right there on the right. Nope. Not alignment.
no?
Where it says
It's like a Q
Yeah, almost like a Q.
next to the A to Z with the down arrow.
Yeah.
Right under view.
Yeah.
I'm trying to find what you guys are sitting there talking about right now.
Okay, now go up.
Go up just a little
Right
bit.
there,
Right there.
there.
Click
Yeah,
on it.
that.
Okay.
So what do you want me to do?
So you're gonna have
Click
to
it.
make sure, click on it. Yeah, so you're gonna have to make sure that all your mutant sequence is as similar as your wild type. So you have like an extra space there, so you're gonna have to try to remove those. So the extra space right there by the CTC, where your start codon's at, you're gonna have to remove that.
See.
So
Hold
go
on.
to the GAA and then click back. The second.
Yeah, so
This
go to
one?
line 2 right before. Yeah, right there and hit delete once.
Yeah,
Mhm.
just once. Okay, and then keep doing that to the CAGG. Just go back and click back once. So you had to do to all of them.
Yeah, just get rid of all those.
Like that?
Just keep doing it. Yeah.
Okay.
It just formatted really weird for some reason.
Yeah.
So
mine-
the alignments didn't align right.
Okay.
And then you're gonna have to do the same with your wild type as well.
So like that. And then if I do that with this
Delete.
one, it should be
At the end of your mutant, delete that last space at the end of your mutant, just in case.
what, this one?
Go up to the end of your mutant one you were just working on and delete that delete that last space there just in case it.
No.
That one's fine. As long as
Oh,
it's
okay.
not
All right.
within the sequence. Yeah.
Okay.
Mhm.
Okay.
And then
And-
do the same for your wild type. Yeah.
Oh, gotcha. Okay. All right. Then I'll just go ahead and redo that after that.
Okay.
Okay. Thank you.
Welcome. Okay, so is everyone good? Hold on, let's just wait for Hydra to get hers all aligned so we can go on and do this all together.
Trying to go as fast as I can. Okay. Okay, so now let's go back. Let me delete this real quick. Delete. And then one. Back. That is, oh, which one did I just do? I put my wild type on first, right?
You can do it however you want. You can put wild type or mutant on top. I did my wild type on top and then the mutant in the bottom.
Hey, Merlin,
Yeah.
does it matter like if we write the word wild,
Does it
like
matter?
when
If
it's
you
all
write-
lined?
Um,
Like when, because I have WT, like it's like WT, and then under that is like mutant, and then like the sequence is for each like line. You get what I'm saying?
would you mind sharing so I can see what you
Yeah,
want?
yeah, sure. Can you see this?
Oh, yeah, that's fine.
Yeah.
Yeah.
Okay.
WT of course stands for wild type, so yeah, that's fine.
Okay. Cool.
It doesn't really matter if I have my flipped right. If mine's like mutants on top.
No, it doesn't matter.
Oh my gosh. I'm getting so confused what I did. TAG. So I have my mutant on top. You guys can keep on going. I'm sitting there. Me.
Don't worry, we're all learning. And
It was fucking me up. Let's go to that. Okay, there. It's right under you now. I have one shot, bro. This guy been after the same fire. Friendly fire with his dark core. Where'd he go? It's up to you, Chow. Wow. What do you mean, Chow? It is, Ale.
What's going on?
Why are you guys hyping him up for, bro?
I think Julian's friends came over. There you go.
What step are we on? Did I miss anything?
The step we're on right now, we're just waiting on Edra to make sure that her alignment's good.
Okay, cool.
And then we'll go into the XPASI translate to get the protein sequence.
So I entered it and now I'm wait. Oh wait, sequence have to be in fast format.
Can I share my screen? I have a question to ask.
Go ahead.
What did I do wrong?
Okay, so I have a question with this. You guys see my Word doc?
Yeah.
So is it these because when if you count, 333. So I don't know because this is 3 and this is 3 because if you count all 3, 3 nucleotide per
Mm.
amino
Yeah.
acid.
Mhm.
This asterisk is at the C. So would I count this as my mutant?
As of right now, because this is our DNA sequence, you wouldn't count the whole.
Oh, would it just
You would just count only the nucleotide.
Oh, okay. So it would just be the C.
Mm-hmm.
Okay. Cool. Thanks.
Welcome.
I'm just waiting. It's running.
Okay.
It still showed up yellow. Okay, hold on. Let me. Okay, so I'm going to paste it. Okay, so let me share it again so you guys can sit there and see because it's still. Okay. Share. Let me do this one instead. So this is my sequence.
Yeah. I don't know. I feel like you're gonna have to talk to the professor then.
Okay.
You made sure that they were both the same. Yeah, it's so weird. And the wild type, the one that you got was the one that we both all got, right? Because even the wild type says there's stop codons, and if we all got the same one, ours would have showed it the same as well.
Well, I know that when he first helped me with it,
Yeah, Ezra, I know what you're talking about because Marilyn helped me on this and it was wrong when he first. I don't know if you're using the one that Marilyn showed you right now.
no,
But
I didn't. This
yeah.
is the one that he did.
Yeah, that one's wrong. So like you should just disregard that one. That wild type is wrong. Yeah.
Okay.
Because I tried using that one too and it showed up like that. And that it showed up green like that.
Oh, gotcha. Okay. So yeah, I'll just go back and do that. Okay. You guys could go ahead while I'm doing that.
Okay.
And then
if you have any questions, you can always stop us.
Okay.
Okay.
Did anybody else have trouble getting theirs to line up once they pasted it onto the Word doc? Because
I
Sorry.
Sorry. Oh, well, what I did right now is that I just switched the margins on the Word doc and I made it
Oh,
narrow, and it worked. So it lined up.
let me do that then. Did you make the size of the paper wider?
Yeah, so like when you click on on layout and you click margins,
Mhm.
there's narrow, and then you just click that, and that's what made it like spread out everything. It's gonna be hard to print, but just to see, it'll line up.
Oh my gosh, wait.
Because mine looks like a little bit funky when it comes, I don't know, like it looks like they shifted.
Did you I didn't copy and paste. I just kept it like the way it looks on the NCBI or the T coffee, because when I tried to do it, like when I copied it to text, it shifted. So
Oh yeah, yeah, I did that. So how did you avoid that?
I just used the I just copied it just like regular. Here, I'll just share my screen and show you. Okay. Oh, I don't know who's sharing their screen, but
Yeah, I'm trying to get out of it and it's not letting me.
okay.
So you can paste both sequences on a .text file, like in FASTA, and then just upload that into Coffee.
Okay.
That's another way you could do it.
Okay.
There you go.
Here it goes.
Okay. Well, I'll just show you how I did it. I'm not sure how he wants it, but this is how I did it. So I just copied it like that.
Because mine looks like that the first time I did it, but now it's all unaligned. And all I'm doing, I'm highlighting all of it and then just pressing copy and paste. And obviously that's not working for mine. I don't know.
Oh, really? Oh, damn. I'm not sure.
It
Maybe-
like it looks funky. Watch.
Yeah.
Mm. Do you
Oh,
see
yeah.
how it's like shifted?
Yeah, maybe try deleting everything and then going back and just copying paste, like delete from the.
Actually, maybe I think it's just the font she's probably using because mine was like that
Oh.
too.
Oh, okay.
So usually the
Oh,
font
you're
for this one is Courier new.
Oh, that's probably why.
And it's at 10.
Oh
yeah,
my
there you
instead of
go.
12.
There you go.
Thank you so much. That is like the
welcome.
silliest mistake that was going to take me an hour to figure out. Okay, there we go. Thank you. Let me stop share.
Okay, so now we're going to do the protein. So let me just share my, there you go. So we're going to get the mutant DNA sequence. And then we're going to go to XPassy translate. And from there, we just insert the DNA sequence of our mutant.
So you just went to XPassy and put your mutant sequence in?
Okay.
Hold on. Let me just share this so I can put this in the chat. Ooh. That did not copy.
Do you want to put like the little caret thing in mutant or just the letters? Oh wait. Or I think you do.
Yeah, you still do.
Like the whole thing? Okay. All right. Let me just delete that.
And now we're going to click compact with no spaces.
This one we inserted both or just one of them?
One, just one at a time.
Just mutant? Okay.
And you put compact,
mutant
no
and then-
spaces, forward, reverse. Okay.
And translate it. And then we're gonna get the frame one.
When you go to the XPassy website, oh, there it is. I see.
And then once you translate, you get the frame 1. And then from there, we're gonna plug that in here.
Okay. Mhm.
And then we're going to do the same for the wild type. So sequence wild type.
Screen. Can you show your XPathY screen again, just to see
Yeah.
if it looks the same?
There you go.
Okay, so that whole first frame, you just copy that.
Yeah, and then you put that into your word
Okay.
because that's your protein sequence. And then once you get the protein sequence for the mutant, we're gonna get the protein sequence of the wild type. So copy that. Go back to XPathY and then just delete that and insert your wild type. And then keep it the same. Translate it. And then you get the frame 1 of the wild type. And then we're going to align this. So sequence, alignment, using tea coffee again. So we're gonna grab this, go back to the tea coffee, and then we need to get our mutant protein. Am I going too fast or?
Yeah, hold up. Hold up.
Sorry.
Hold on. Click. So you didn't go, you didn't use, so the, okay. So you're gonna put both of them in 2 coffee?
Yeah, for the protein sequence.
Okay. Tea coffee.
Just, do we click on the middle of tea coffee again, or do we go to the protein
Yeah,
one?
you the tea coffee one. Because that one aligns DNA, RNA, or proteins.
Okay, my alignment came out pink.
Okay, good.
That's good.
So using your DNA sequences of the mutant and wild type, Edra, you're going to use XPathY. So you're gonna do these one by one.
Okay.
And when you translate them, you're gonna get the F prime to 3 prime frame 1. So this is what it should look like. So this is the protein for the mutant and then the protein sequence for the wild.
Do we just leave default settings on this?
A minute.
We want residue on 60 again though, right?
Yeah.
For the length, does it matter or has it be 60 again?
You could do 60 again. So we're going to copy this, and then we're going to insert it into our Word. Wait. And then if you guys want, you guys can highlight which one is your mutant. Oops, not that.
Should the XPath translate sequences be pink as well on the Word doc?
Pink as well?
Yeah, like because the sequence alignment is pink.
Like this?
Yeah, like I copied that and straight onto the Word doc, but it's like pink. Yours isn't pink. Does that matter?
Like highlighted? I don't think so. Unless you want to remove the highlight. You want to?
If it's not a problem, it's fine.
Yeah, I don't think it should be a problem because I think it's just highlighted.
Yeah.
I think you
Yeah,
can
mine does the
remove
same
that.
thing, but
Cool.
Okay, so these are our mutants. And then the last step is that we're to create a paired nucleotide and a protein sequence. So we can grab your DNA sequence again for your mutant. See that? And then we're to go to XPath, and then we're to paste that. And then you're gonna want to include nucleotide sequence.
okay. Hold up. So good. You didn't get your mutant, you said, or
This one's mutant, yeah,
okay.
for XPath again. And then we're gonna the output format, it's gonna also include the nucleotide sequence. If you translate it, you're gonna get all this.
Include. Okay.
Okay, and then you're going to
Those yellow highlights, where did that come from? I don't have that.
Oh, that I did
Yeah, I
myself,
did it.
just to make
Yeah.
sure to know that those are my mutants.
So on the XPath, when I go into for the protein sequence, is there anything that I need to click on?
For the XPath, which your mutant DNA sequence, you're gonna click on includes nucleotide sequence.
And that's for the first, or is this the second one?
For
Time.
this is the second time. The first one you should have gotten only your protein.
Right.
Mhm.
Okay, so but there's nothing that I need to click on, just it just being let me see, hold on. The only thing that is on there, like compact, is that
Oh, no, you're gonna do the include nucleotide sequence for the output format.
for both of them?
Or wait, which step are you on? For the
I'm
protein one?
on the protein one, yeah.
Oh, okay, for that one, yeah, you're gonna put compact then.
Compact. Okay, okay, okay.
Yeah.
All right.
Marley,
Yeah.
do you think you can take away recording privilege and give it back to me again, because I got kicked out of my Zoom?
Yeah, hold on. I think I'm gonna have to.
Wait, how did you find the identification of the mutant?
From the protein sequence alignment using tea coffee. So you're gonna align your protein sequence for your mutant and your protein sequence of your wild type, and anything without a little asterisk is gonna be your mutant.
Oh, okay.
Okay, so who was it that said that needed the recording?
Me, was that?
Okay, there you go.
Thank you.
Welcome.
Can I share my screen because I'm still confused? I aligned it, but I don't know how you found the identification part.
Oh, yeah.
Okay, so this is mine right here. Can you guys see the Word doc?
Yeah.
Yeah, so I found
anything without an asterisk. Can you
So the rows, you need to line them up like
Yeah.
on the left side. I had to delete like on the top where it says 1 and 1 on the left.
right here?
Go up a little. Yeah, line those ones up. Yeah, delete like one space there.
Okay.
Keep going there. Now stuff should start lining up.
Okay, so I'm gonna do the same thing here.
I think you
That's
should.
what I had to do to get mine to line up.
Yeah, I think you should put the cons right below the word mutant too, so it can like so you can see better with your asterisk.
Wait, so then I mean, so like, well, this is kind of confusing.
Like the cons, I think. Well, to me, I just say like I think it's just better you just delete it so it can line under the word mutant, the word cons.
Yeah, what about it?
Like to me, it's just to me it's better just to see where the asterisk is actually hidden. But I mean, you can still see it.
Yeah. So all the numbers need to line up essentially along that left edge.
Okay. Looks like I have asterisks
when you
for
look at
all of my things though.
No, you don't. So I don't know if you can.
Oh, like right here? Like the 162 should line up with the 162, right?
Yeah. I think it's just the font of your text. So try to do the Courier New.
Okay.
Sometimes it for the whole thing, yeah. So.
Which one?
It's Courier New. So go a little bit more lower. Oh, that one right there, the new one.
This one?
No,
Not that one?
The one.
Oops.
Yeah.
Okay. Go back.
So I have one gap that doesn't have an asterisk, and then another one has a colon.
That one, he said not to worry about that one, and then change that to 10, your size font.
Okay.
Hopefully that should help.
Okay.
It's still aligned a little bit off.
So should I?
Those, hold on. Can you check to see if there's extra spaces just to see if there is?
Like where?
Using the symbol that Edgar used to see if there's like extra spaces there. So
How did you? What symbol?
you know where the AZ and the down arrow is that the one next to it that shows if there's extra like spaces.
This one?
No, the other one next to it. on the right.
Oh, paragraph?
Mhm. So maybe try deleting those little spaces.
Is it these little dots
So
right
you
here?
just, so I just fixed all this. You're just gonna have to manually go and line up each pair of rows with the space, like line up the ones, line up the 55s, and all the rows will start lining up.
Okay.
Yeah, like the mutant, the one. just
Yeah, like
oh yeah, like that.
delete back until the, there you go. See how the letters are matching now?
And then
Yeah.
the cons do that too.
Do that for each row
Astra has
and
to,
the con also.
yeah, okay.
Yeah, there you go. All the ones are matching now. Do that for all your rows.
Okay. This 162 right here, should it be up here?
Don't worry, just
do the
move the
109.
109 back.
Yeah.
Okay.
And then, so there's your mutant there, it looks like, the KA.
Mm-hmm.
The KA.
Yeah,
It's the circle,
the little
the
circle,
circle,
within the asterisk.
yeah.
Mm,
On that row, the 109 row with the circle instead of an asterisk.
oh, this one?
Mm-hmm.
Yeah, that's your mutant. So straight up is KA, there's your mutant.
So yeah, that's my only mutant then.
No, I think you have a second one, the AT as well.
T.
The one that has like a colon.
Oh, this one?
Yeah. I think that's probably your 144 mutant, the one that he says that some student has 2 mutant.
Yeah.
Cool. Okay, so
So
then
if we have a colon, that's another mutant.
Yes.
Yeah, that's another mutant, but I remember from him talking in class is that one we're not gonna worry. So that one, he said basically think of that as like almost like a wild type. The one that we're trying to focus on is the one without anything. So the K and the A for Jasmine.
okay, so then it would just be the K and the A for me.
Mm-hmm.
And then how did,
you still have to mention that you have it though.
oh, I do? Okay, so I
Yeah.
should still highlight it?
Yeah. But the one that the mutant that we're gonna worry about is the K and A.
A.
Mm-hmm.
Okay, and then how did you get the I think it was the identification one?
So you're gonna get your DNA sequence for mutant and then go back to XLC translate. And you're going to, instead of the compact one, you're gonna do the include nucleotide sequence.
So I go to XPASI back,
Mm-hmm.
and then I put in XPASI, I put in which one?
The mutant DNA sequence.
Okay.
Do we, when we put that in, are we having the in, or do we have that with the changed?
The change?
Because the mutant, remember we found an in and we changed it.
Mm-hmm.
So when we copy it into XPASI, do we leave it with the in or with the changed letter?
With the changed letter.
Do you think we have to report that too, like for Christian, where he had to report that he manually had to read the peak for that?
Yeah. Because to be honest, I don't know if it's correct. Just in case he tells you like, yeah, there's an error within your sequence, just say I had to manually analyze my DNA sequence.
Can you show that step again, where this last T coffee step?
Yeah. Hold on, let me share it. So T coffee. So it was the multi-alignment. So on my Word docs, the multi-alignment. And I highlighted my mutant, which was the E to A. And then what's it called? And then the A and T. And from there, I got my DNA sequence of the mutant. So I copied that. I went to XPOSSE and then I inserted that. And then I clicked the include nucleotide sequence and then translated it.
Okay. That's what
And then
I
you're going
was-
to get this. So you copy this whole thing for frame 1. And then that's how you're going to identify your mutant. I say highlight because then you're gonna have to highlight them here. So you know which ones.
Okay, so I have a question.
Mhm.
So for the tea coffee one, the one we just did previously, so I don't have like a mutant you per se, because mine has all the all the little stars, except one, and it has the colon. But you said that one doesn't count, so I'm like, oh shoot.
If that's your only mutant, that one's your mutant then.
Oh, okay.
Yeah.
And I only have one, so that's normal, right?
If that's your mutant, then that's your mutant. The only reason why for us it doesn't count is because we have 2.
Okay.
Yeah, he said in class that if this is your only one, then that one's your mutant. But if you have 2, then you should worry more on this one than that one.
Got it. All right, cool. Cause I was confused. I'm like, shoot. Okay. Thank you.
I'm sorry. I still don't know how to find the identification of the mutant. So I go to tea coffee, right? Can I share my screen again?
Yeah, go ahead.
Okay. So where's tea coffee at?
Tea coffee is to align your protein sequences.
Okay, so I'm in the XPASSY right now, and this is the one for my previously. But if I want to find, okay, so first you went to tea coffee, right?
Tea coffee was just for the alignment to see which one your mutant was.
Okay, so I found my mutant, and then where did you go from there to find the identification, that long one?
So you're gonna get your DNA sequence of the mutant, just that one.
Okay, so this one, right?
No, that one's protein. The one that we started with in the beginning. Yeah, the very beginning.
Oh, the beginning.
Wait. That sequence. Yeah, you're gonna copy that. And then you're gonna go to XPOSSE.
Okay. And then I'm gonna paste it.
And you're gonna paste it, and then you're gonna click, instead of compact, you're gonna click include nucleotide sequence.
Okay. And then forward, reverse, and standard, right?
Yeah, leave those by itself. And just click translate, and then the first one, which is F prime to 3 prime frame 1, you're going to copy that one.
Perfect, thank you.
Welcome. Okay, so from here, we discover that these were our mutants, so we highlight those. And so now we just need to label them. So it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So there's 20 here, 40 here, 60, 80.
Oh, shoot.
Yeah, my formatting came out all weird. Yours pasted nice and neat.
Yeah, man. Yeah.
Oh, I had to fix the font for this one. For this one, I did the Courier New, and then it's 9 instead of 10. So 20. I think that's 130 to A, and the A, I think it's 144, I believe. 240.
Yeah, Courier New 9 works.
145. There you go.
Yeah, just to let you guys know, you know how the position, like how Maryland have 145, but he said don't count 145, count 144.
Oh, because of the
Yeah, it's just the same. I think that was 129.
yeah. So that should be 129. Because he said the start codon. I think I remember. So it starts on baleen.
Yeah, correct.
Would you still label it 20, 40, 60, or does it become 19?
I still label it 20, 40 because it's easier for me to find the exact position and just -1.
Okay.
Okay, I have a question. How would you find your mutant in the ID of like in the bottom one?
And the bottom one?
Yeah, like
So for my protein sequence alignment, I highlighted which ones. And so I tried looking for the sequence that looks similar to that one. So it's SAKV. So SKV.
oh, I see what you mean. Oh, okay, when based off letters. Right.
Oh, I didn't even think of doing that.
Yeah, I mean, either I'm like, wait, I don't know actually.
And then save this because you're gonna have to use this for your presentation and your report.
Okay, ITQ.
Wait, can you say that again, Merlin, about the identification of the mutant?
On what part, Ty?
This part right here, the one you're at. Like, once you highlight the letters, like, how do you find it in the?
Okay, so how I found my mutant is I try looking up at least 4 of the nucleotides. So it's SAKV, and so I start looking around here because it's a little bit more nicer, and it's SAKV right here.
Okay, cool.
Thank
you.
And it's the same for this one. It's GRIT.
And you're just looking at the mutant line, right?
Yeah, just this is only mutant.
Why did you name it? I understand the 129A part, but why the E part? E129A over A144T.
So E, because in normal wild type, the CRP gene, it's an E. But our mutant, that E is an A. So E is saying, like, this is what it was before, and this is where the position's at, and in our mutants A.
I cannot find this thing.
Did someone say they can't find theirs?
No, I can't find it. Like the
Want to share your screen so we can help you?
I'm not finding mine.
It's like, yeah.
Okay, if you guys want to share your screen, I can help you guys find it.
Oh, you can, let's see. So my mutant is this one. The ITQ and
Okay, so hold on.
then I'm sorry. So I got this one. I got the ITQ, the mutant. I got it. But the wild type.
Oh, this one won't be on here. So
oh shoot. Okay.
Yeah.
Because okay. Because I saw you guys have 2, but I forgot you guys had okay.
Yeah. So that one's your mutant. So you have the AT. So yours is gonna be A144T.
Wouldn't he have another one, or can you just have one MAA 144T?
I think he only has that one. See,
scroll up. Yeah.
Because look, everything is filled up.
Yeah.
Except this one.
Interesting. Okay.
That's his only one. Yeah.
So it would be what would it be again? It would be
A144T.
A44T.
Yeah, A144.
Julian, I honestly, I would ask him too, like after now that you're gonna have it done by Tuesday, I would ask him because that shift is supposed to be just like a mechanism he used.
Yes, I see what you mean. Yeah.
I think he talked to you about it in front of us, right?
No, I don't know what you mean by the A44T. I will tell him though.
Yeah, just to make sure, like
it's only there too.
Yeah, because you're doing the steps right. So if it was like his mistake that he didn't put a mutation in, then so that he knows.
All right, I'm gonna tell him that I deserve extra credit. Nah, I'm kidding.
For him messing up.
Can I share mine too?
Oh yeah.
I'm not seeing it.
Back up.
Okay. So what's your mutant? The A G L F. Okay.
So my mutant is FAE, right? Or I mean, it's an A, but I'm looking for FAE down here
Yeah.
somewhere.
F A.
Where do you use the
I think
find?
it's right here. Hold on. I think it's this.
Oh, yeah, you found it.
There you go.
I'm just bad at word searches. Okay, cool.
Okay, so just highlight that one and then the other one. Hold on.
For ITQ.
Right here.
Awesome. Thank you.
Welcome.
So we subtract the number that we get by 1, right?
Yes, correct.
Okay. Okay, just making sure.
Can I share mine too? Because I can't find mine.
Yeah, go ahead. Actually, I'm really good at word searches. So I don't mind helping you guys find them. Let's see. Annotate.
So yeah, I have the K and the A and then the A and the T right here, but I can't find them down here.
Right here.
Oh,
And they're right here. I think we have the
Oh, wait.
same one.
really?
Yeah. I think. Let's see. Hold on.
So how do we know what our mutant is?
How do you know what
Like,
mutant it is?
You were able to name what the name of the mutant was?
The mutant, you have to use the numbers. So for hers, it's 120, and then how many are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So 131, 130.
Oh, you just find the position that's located?
Yeah, because
Okay.
when we're finding the position, we don't count this one. So you subtract.
Okay, and then what's the naming scheme of it? How do you actually phrase the
So
name?
from the scheme, it's going to be the wild type, the original nucleotide, the position of where that nucleotide is, and then what it changed to in your mutant.
Okay.
So for, I think Jasmine, right? Yeah. So yours would be
Yes.
K130A.
Wait, it would be A130A?
It's K30
Oh,
and then A. Because I think this is 31 right here. 30, yeah, 31, so it would be 30 since you
so
subtract.
then it would be over, and then it would be
The
what about my A and the T?
that one would be A144.
A144T.
Yeah.
Okay.
Can we double check mine just to make sure if you
Go ahead.
Okay, cool. So okay. So this is what I got. And then so here's my highlighted mutant. And then I put it right here. And
Yeah,
this is what
that one.
yeah. So
Okay. So
yeah.
that one was 31, 32, 33, 34. Yours is gonna be, was it a V? V
It was N, I think.
N? Okay.
Yeah, N.
Oh, yours is an N?
Yeah.
You didn't. Oh, you have N's.
Yeah.
Yeah, you needed to manually
Oh, okay.
switch those. Yeah.
It's on my wild type. So
And nucleotides abbreviation though.
Oh yeah, it is. Oh, We're doing protein. Sorry, I thought it was a DNA. Yeah, so you're right. So it's N. Let's see. Hers was 31 right here. 32, 33, 34, 133. Yeah, you're right. And then the A44. Yeah.
Carlos,
Yeah.
can you scroll up a little bit?
Yeah.
Oh, okay, so your okay, so A to N, so be N to A. Okay, yeah, I'm just checking because you have the while on the bottom. Yeah.
Yeah. Thanks. Yeah. Okay.
So it's in 130 whatever A.
Mm-hmm.
Okay. Cool.
So we only highlight one of our mutants. We don't highlight the other one.
You have both, and you're gonna identify both, but for this lab report or lab project, we're only focusing on the
AT.
Not the AT, the other one, whichever nucleo amino acid you have that switched to alanine.
Because okay, so when I saw yours, you only highlighted the one with the semicolon, which was the AT, right?
No, I also highlighted the one from wild type from E to A because mine.
Oh, you also highlighted that one, so you highlighted both?
Mm-hmm.
Okay.
Do y'all know where we can find the information for the presentation? Like this.
It should be on Canvas.
Yeah.
But basically what we're doing here is what you're gonna put in your lab presentation. Because the lab presentation is basically what you have been doing for your report for lab. So it's all the DNA sequence, all the protein like analysis. So we're gonna use like Chimera or the Swiss PDB viewer, and it would be also like say your how you interpreted your dishes.
Oh, okay.
So we're just going to kind of summarize what we just put on our Word docs in a PowerPoint and then add in the stuff about the colored dishes.
Yeah. Mind you, it has to be 15 slides too. So.
Jeez.
Question.
Oh, yeah.
The Word doc, the written, I guess, part, how are you supposed to write it? It's just
for
like,
the Word doc, you can keep it like this. You just put figure 1, what you did.
Like how he had it on the Word doc, right?
Mm-hmm.
Oh, okay.
Okay, and I think we're done with this, unless anyone else has any more questions.
Could you check mine real quick?
Yeah.
Please.
Mm-hmm. Okay, so can we see your protein alignment? So, Ty, your con, it's off by 1.
It's off?
Yeah, it's off. So you're gonna have to delete a space between the con and then the 109.
The con and the 109. Oh, here?
Where the 109 is? Oh yeah, right there. See? So it's not your Q's that has the mutation. It's the A and the T right here.
Okay.
Yeah.
Got it.
What's the due date for the PowerPoint again?
Not this Tuesday, next Tuesday.
Okay, cool.
Mm-hmm.
Are we meeting again before then or no?
Yeah, if you guys want to. If you guys want to like talk about anything else.
So tonight we're doing this and we're doing Chimera.
Yeah, unless you guys are like, yeah, I'm done. This has been too much. But if you guys want to go over Chimera right now, it's gonna be like a whole nother hour probably. Unless you guys need to do something.
Should we split it for another day? I'm okay with staying an hour, but I don't know if you guys have other assignments for other classes.
Yeah.
I use Sunday as my catch up. That's the only reason I'm asking.
So you're gonna have to fix the name for your mutant. So it's gonna be A144T. Ty.
A144T.
Mm-hmm.
Are my names right? Is the first one right?
Yeah, the first one's right.
Like the way it's named?
Mhm.
E5070. Okay, cool. Thank you.
But yeah, if you guys want to split up the Chimera, then we can. If you guys don't want to, I can continue going.
I'm down to just keep going and knock it out. You don't.
Can I share again one more time? I just
Yeah,
wanna
go
make
ahead.
sure it's right. Okay. So I
Hard to
got
keep going.
I got
This is like
this.
trying to
right here. But is this correct? K130A?
Okay, so
Because I'm confused because you said K130A at first, but I don't know where you got that from.
yeah, so okay, hold on. Let me annotate this. Okay, so right here, that's 20, 40, 60, 80, 100, 120. And so right here, it's 30. So that's 30, right? You're at
Yeah.
31. When we're identifying, we don't include the methionine here. We start here. So if yours is at 31, you subtract the 1 because we don't include the methionine. So it's gonna be 30.
So am I supposed to highlight my K or no? My mutant, my K?
No, because your mutant's an A, it's not a K. But yeah, you can
Oh,
you're highlighting it.
oh, I see. Okay.
Yeah.
So I just leave those two highlighted, just the A and the T then.
Yeah.
Okay. Thank you.
Welcome. Okay. So no objections to doing Chimera?
Can I share my screen real quick?
Yeah, go ahead. Okay, this is your wild type protein. Is the mutant one on top? Yeah. Okay. So what were your protein? Okay, that one's DNA. For the protein sequence, did you align them? Hold on. I think, Idra, are you on mute?
I think she lagged out.
Oh, yeah. Ooh. Wait. Ow. Ooh. Hold on. Edra? Yeah, I think she is. Okay, well, I'm gonna stop your sharing. Did I stop the sharing, guys?
I still see.
associate.
Oh yeah, it's not letting me. I think it was not. I don't know if she.
What if you enable or disable, I mean, sharing?
Hold on.
That or you'll have to kick her temporarily.
No. Yeah, I have to. Oh, I don't want to kick her out though. But let's see.
Just give her like one more minute and see if she reconnects it.
Yeah, if you kick
Oh,
her out,
there you go.
group me and let her know.
Should we do a vote on here if whoever wants to keep going with Chimera or not? Because I feel like we've had a long day already.
Or we can also like Merlin, where did you put the recording last time? I remember somebody asked you for it and you were able to give it to
I put it on YouTube. I actually didn't have the distance on that one because that one I didn't know yet. But if you guys feel like you're done, I can make like a separate recording just by myself and like post it up and you guys can see it.
Or I could,
or if you guys want to like know like step by step, when are you guys open? I'm basically open like every day of the week.
I mean, I'm down to keep going right now. I'm trying to knock it out.
we can post a recording for people who can't keep going.
Yeah, let's do that. We'll just record it.
Yeah, I still have my recording and I can post it on like YouTube and then send you guys the link like I did for Ty for the lecture.
Yeah, that would be awesome. That way, like whoever can stay and whoever can't, and then we'll like it helps to see other people problem solve.
Mm-hmm. So yeah, if that's an issue that you guys can't stay or you're like it's been a long time, I have other stuff to do, then you guys are more than welcome to leave. I will keep recording, and whoever wants to knock it out right now will knock it out.
All right.
Audra said her laptop died. Hold on, let me text the group.
You know, I have a question that I'm kind of a little bit conflicted on.
Yeah, what's your question?
So you know how he keeps saying that the active CRP, we're not we're not supposed to see anything interacting with our molecule or our chains. So I'm confused because I did the practice one and I did, you know, the 2 Armstrong and the 4 Armstrong, and I saw molecules so interacting with the active CRP. So I'm kind of conflicted because he keeps saying we're not supposed to see anything. But on the exam, he also said, you know how the exam he gave us a question about the active CRP as well, like name four proteins are interacting with the active CRP. So that's why I'm like, are we not supposed to see anything, or are we supposed to see something, or what's going on?
Maybe there is something. I think I remember him saying anything within 2.5 to 4 Armstrong, it's considered interacting. So I think it is. For the lab project, is it both the inactive form and the active, or is it just one?
That's where I got confused because on his example format thing, he had both active and inactive, but he only posted like the Chimera or like PDV viewer file for the inactive form. So that's where I got confused, if that makes sense.
Yeah, the one that is active, I think the name of that one is 1-CGP, and the inactive is 2-WC2.
Yeah.
When he gave us on the PowerPoint, he gave us practice once before he gave us our actual data, and I was looking at the active of mine that he gave, and then it had, you know, other proteins, R chains interacting with my R chain. So I was kind of confused because I was like, I thought he said that we shouldn't see anything.
So you did your mutant to the file, the 1-CPG file, and you found residues? Or are you talking about that first example that we did?
The first example, I looked at the mutant, I don't know, the mutant or the data that not the actual data, but the one he gave us to practice on. And I looked at it in the active form and I saw molecules interacting, and I did it in inactive. I also saw molecules. So I'm like a little bit confused.
Yeah. I guess we'll figure it out right now.
Yeah. Let me check because I don't know if we're supposed to do both active and inactive.
Yeah, that's where I got confused on, because on his report he has both. So I got worried because I couldn't find anything for that active.
I had went to his office hours and I asked him because I was trying to work on my paper and I wasn't sure either when I got to that part. And he said we are supposed to do both. That the inactive one is the one that he posted in Canvas, the 2W whatever model.
Mm-hmm.
And then he said the active form we have to get from PDB, and the name that you'll type in is 1CGP, and
Oh,
that'll be the
so
active form.
did you find anything?
Yeah, or
Oh.
interact, or like when I typed it in PDB.
Oh, no. Did you find any active or interacting partners in the active form?
I had
Or you
like
haven't gotten?
started it, but I couldn't find the distance, so I
Oh,
don't
okay.
know, like if it's close enough. That was the one thing I was having trouble with. Is there? It doesn't look like in the inactive one that he posted.
Mhm.
There's like one or two that look really close to it, and then on the active one, they're a farther away. So I don't know if
Oh, okay.
consider
Okay.
like interacting or not.
Okay, so we'll do them right now. Okay, so which one do you guys want to start with? Do you guys want to start with inactive or active?
We could do inactive since you know how he removed the stuff, so it's less chains versus the active have like A through F. That's a lot of chains. So you could do inactive first.
Okay. So let me just- Hold on, I think it's on my desktop. There you go. Okay. So let me screen share. Okay. So we're actually gonna move. Okay. So you guys see this?
Yeah.
Okay. So this is our inactive protein.
So did you open that from the one that's posted?
Yeah, I opened this one from the one from Canvas. Okay.
Where's it at? The Canvas one?
So that one should be in modules. There's a YouTube video on top of it, and then it should say the 2WR2 model.
Oh, the model one thing?
Mm-hmm.
Okay.
Okay, and then hold on. Just make sure you know which one's your mutant because we're gonna have to use that one. So 129. Okay. So
Can we open that with PDB viewer?
yes, or Chimera.
For
Gotta download
this
it.
one, we're only
I guess-
viewing the first mutation, not the second A144T, right?
Not the second, only the first one. Mhm.
Thank you.
Unless the A414T is the only mutant you have, then you're working with that one. Hold on.
Where did you download Chimera again? Because I know I downloaded it the first time, and then for some reason I don't have it anymore.
Check your downloads file. It's probably
Yeah.
in there.
It deletes it on Mac. Well, on my MacBook, it
shoot.
deletes it. So just go to, if you go to the what is it called? The Launchpad. I don't know. I think that's what it's called. But if you go there, you should be able to find it. But just like scroll to the right.
Sorry, guys. I'm gonna be taking screenshots because
Hold on. No, I don't have it.
Did you try to I go to Finder and then I go to download, I search for it on the search bar. You have Mac?
I do.
Okay, and then I go on the search bar and just look up Chimera.
You go to Finder and then where?
I on the search bar in the upper right corner,
Oh,
like where Merlin's showing you right now
oh,
on the
Oh.
top.
Okay.
And then I pull it out onto my desktop, and that usually makes it work.
So
Wait, do we use Chimera too if we had PC?
I think it used PP. What is it like? A
PDB.
different.
I just used Chimera and it worked fine on PC. I just got the exact same picture that she has up.
If you want to try for your PC, if you can get Chimera, if you want to, you should.
Did you just type in like download Chimera on Google to download it?
Let me look really.
I can't find it.
I did Chimera structure viewer,
Okay, Chimera.
and then there's an option to download Chimera.
Okay, I see.
So Chimera. So for Julian, you can do the.
Oh, and I drag it out, right?
And then just
This
download
did?
so this
You
one,
drag it out.
and then you click on this one.
Yeah, if you have PC, then you would do the Microsoft window.
So this is the inactive.
This is inactive. Take pictures of it because you're going to need it for your report.
Christian, how did you download Chimera? Because I went to, because I put it in Google and then,
I'll put the link in chat here.
okay.
There's the link, and then you'll just pick if you're Mac or Windows.
Okay.
Okay, give me a second. I think it's like verifying Chimera or something like that on my laptop.
So this, what is this called? The inactive.
This is your 2WC2. This is the inactive form of CRP.
Okay, I opened it.
Is it up?
Yeah.
Okay, and do you have your model?
Yes, that came up.
Okay.
It's kind of, oh, yeah, it's kind of way too zoomed in. Okay.
Okay.
All right.
And so is everyone good, or should we wait on everyone that's downloading right now?
What's the thing you told us to take a picture of again?
Take a picture of this if you're
Oh.
phone, screenshot, whatever,
Okay.
because you're gonna need these for your presentation.
Okay.
I'm still downloading right now. After I download, where do I go?
After you download,
You're just going to install Chimera, and then you'll open that PDB file that is on Canvas.
Which one?
oh, hold on. It's the one in modules. So right here, where it's
Yeah.
structure analysis, this is the one that we're using.
So so you went to Chimera and how did you open that one?
You just click on it and then it says download and then it should say like open with.
Hopefully it should open with Chimera. I had to go into Chimera and go to open and then navigate to where I had downloaded that.
Okay, I'll try it.
This, I mean, scientists made it. It's not the best software. It's pretty clunky.
And we had to learn how to use it because our professor did it.
Yeah, you can tell that it wasn't a software developer that made it. It was some scientists that learned how to make software so that they could make this.
Okay. So just whenever you guys are ready, we can start doing The structure analysis.
I'm just installing it right now. It's like extracting files.
Yeah, that's fine.
So technically everything has to be done by next Tuesday, right?
Yes. Not everything, just the lab presentation. Sorry, not everything. Just
Okay.
the
But we-
15 slides that we're doing.
yeah. Okay.
Yeah, I think May 5th is when we should be done with the report.
So all of our presentations are pretty much gonna look the same, right?
Yeah, really the only different step is just your mutant, and this is gonna be very different because everyone's gonna work with their mutant at this point now.
Mm-hmm.
We should just come up with a template and everybody use the same template.
So he grades on that scale, same.
Yeah, I mean, he doesn't care about creativity. He just wants all the same stuff. So then we all know we're on the same playing field.
That's actually pretty smart. Down with that.
I don't know, but I feel like he's gonna be like, well, did you guys just copy each other?
We worked on it together and we have proof.
We worked. Yeah, there's nothing wrong with that.
Well, that's true.
Yeah, we even have like the
It's not like
recording
you
of this
can
video we're working on
cheat.
together.
Yeah, there's no way to like cheat on this. You just all work together to figure it
That is
I
true.
had him last semester, and I know like we had it was kind of like the same. It was a whole presentation, and I know like all the presentation sound exactly the same, especially because you're using the same images. Sometimes it's like images that he provides, especially for like the actual scientific part that you do in lab. Like he's gonna give you all those pictures, and like every single presentation was like a repeat of the other. The only thing that was different was the part of like your oh, like this is my mutation, like the beginning part, you know?
Yeah. Was he harsh in grading?
No, like he was pretty easy. Like I don't know, this semester he is a little different. Like before, I feel
I'm
like
pretty sure I was looking at the rubric when he showed it for this, and if you did poor on your writing and poor on your data, you got a 70%.
Correct. That
I'll
is
need to
true.
pull the rubric up again, but it looks like if you just did horrible on the whole thing, you got a C.
Like the lowest you can get is a C. Yeah, I saw that too.
So in a way, is that saying that a C is guaranteed?
Yeah, pretty much.
And then even then, he did say that at least 6 people are gonna get an A.
So that means everyone's gonna pass the class, and that's something to be
Yeah.
happy about.
Well, in that case, why are we doing this? Nah, I'm kidding. Nah.
This is a guaranteed C.
Shit, nah, I'm kidding. Alrighty.
That's a whole senioritis mood.
For real. I'm like,
Honestly, if what is it, parasitology was available, I would have taken that instead of this class. But that class, the professor retired, so it wasn't available. So I had no choice but to take this class.
I only took it because it sounded cool. And then,
I mean, this is pretty like advanced, but I feel like it's not as bad as it could be. At least we're able to use our notes during the exam.
yeah, I don't think the material, like the class itself, is hard. It's just like his lectures are kind of confusing sometimes. You know what I mean? It just takes a little bit extra more work to actually understand what he's saying, but it's pretty like straightforward, the material.
You guys are being very generous.
Okay.
No, trust me. I did very bad on the test, but I know my, but at the same time.
I did okay. I went to talk to him. I was frustrated with myself. He even told me, he was like, I don't know how you got the hard ones correct, but the easier ones wrong.
I mean, you already got an A in this class. Like he's already put your grades in and submitted you for graduation.
100%.
I don't even know. I feel like if enough people start talking to him. You might even curve it even more. Are we all ready, or do we still need to wait on installing?
I'm ready.
I finished installing. I'm just trying to like find it, I guess, on my laptop.
Find
Do you have Mac? Do you have a PC or a Mac?
I have a PC.
Does anybody who have a PC can explain to him? I wouldn't know for PC.
I think
So when you go to open, it's like I can't even explain how bad. Like they didn't do it very easy. I had to go and find where the download was and find the actual name of the file path and just copy and paste that into the open.
Do you want
Okay, let
to
me try it.
sharing so you can like show him?
Yeah, give me.
If you can.
Let me see if I
Yeah.
can. So here, I'll just type it like in chat. Like I named the file that we downloaded inactive.pdb. So I had to actually go find where you downloaded it, find the file path, and just paste that into where you go to open it. You can't just go to open and then like navigate through folders. I don't know why they made it super difficult.
Okay.
So for me, all I had to do was open up the PDB viewer. Do you know how to open it up, or are you trying to upload the 2WC2 file?
Like I found the camera application. I'm just trying to open the.
Oh, you did Chimera? Okay, never mind. Sorry.
Let's see.
Ty, do you
Yeah,
have
just see if you can find your file path.
Yeah, because I don't use, I have a Mac, but I also use PC here and there. I know that they also, if you go to my documents or sometime in the folder on your, I think, left side column, there is a little tab that says download. And then you can just click on that and everything that you download drops into that download folder.
Hmm.
So you can, if you go to the file you downloaded and right click it and go to properties, it'll tell you the file path and you can just copy that.
Okay. Let me try that. Let me try that.
That's kind of easier. You'll need to add the name of the file to the end of that file path. Like if it's in desktop, you're going to need to add a slash and then the name of the file. If you paste that file path into chat, I can change it if you have trouble.
Okay, let me copy this. Not pasting. You guys can probably just start because I can always watch the video later. I don't want to hold anyone up.
I'm literally having the same issue. I can't find. I can't
Yeah,
find.
like I have the application open. It just won't let me open the inactive thing from Canvas.
Okay, so go to the file you downloaded it
Yeah.
and right click it and go to properties.
Yeah.
And it should show you the file path.
What do you mean?
Like it'll say where it says location, it'll say
Right.
like the drive and all that.
So we copy the location.
Copy the location,
Okay.
but you're going to need to add one more slash to the end of that, and then whatever the name of the file is.
Okay.
I renamed mine to inactive.pdb. So, but I think it's something else when you download it. If you look in chat, you can see how mine's kind of formatted.
Yeah, the
okay.
original file name is 2WC2, I think.
Okay, so after that location, add one more slash like I did and type that file name in. And then you'll copy that whole location into the open of Chimera.
Fine. Okay.
If you need, just paste your location in chat and I'll add it to the, like I'll make the link for you.
Okay.
Yeah, I finally got it. I just copied the properties slash and then the name .pdb.
Yeah.
Oh, it says it's a PA file.
A what?
Well, I looked at the properties. It says the file is a PDB file. I don't know.
Yeah, so after
It is.
yeah, so after the name, put .pdb and it should open.
If you're having trouble, just copy paste your location of that file into chat and I'll do it for you.
Let me try to do that.
Okay.
Were you on it, or is that wrong?
It doesn't look quite right, but I think I can fix it.
Okay, because I didn't add the C colon slash, but that's
Yeah,
like
we
all the
want all of that. If you copy that whole thing, that's what we want.
Okay.
I was just going to add it. I assumed it was the C drive, but if you copy the whole thing, yeah, perfect.
Because I'm trying to open it with Chimera, so I don't know how to do that.
Okay, there. Now copy that, and when you click on open and Chimera, just paste that and hit enter.
Okay, copy this and then go to Chimera.
Yeah, go to Chimera and click open. And where it says file name, just paste that there and click open.
Oh, cool.
Sweet.
All right, I got it
Good
open.
work. Okay,
Thank you.
so now we can start.
Let me take a picture.
Yeah, take pictures along the way, screenshot for your projects. Okay, so from here, we're gonna go to favorites, and we're gonna go to sequence. For sequence, we're gonna click on A1 just to focus on that one for right now. We're gonna click on A, and from here, we're going to highlight whatever was your residue. So,
Wait, okay, hold up.
Oh,
Click on
sorry.
NA.
Let's start over.
She's like, wait, no.
Sorry. Okay, so we're gonna go to favorite right here,
Okay.
or we're gonna go
Yeah.
to sequence.
Okay.
Oh, sequence. Okay.
From here, both chain and chain B are highlighted. So we're just gonna click on chain A to
Okay.
work for that with that one right now. So we're gonna click on that one and we're gonna click
Okay.
on show. And then from here, whatever your mutation was, you're gonna try to look for it.
What mutation?
The mutation that we had.
On
Mutation.
Like mine was E77A, that mutant.
what if I have one
E7.
the 144 thing, Mindy?
The 144?
Yeah, that's the one I had, right?
Oh, nope, I did not mean to click that. Your A144 is gonna be
Wait,
right
did
here.
all have 144?
No, we're gonna focus on the one. If you have two type, we're only gonna focus on the first one. We're not gonna focus for the 144. But because Julian only had one, he has to focus on that 144.
And mine was
Okay.
144 right? 8.
So we're gonna try to look for it. So mine was E.
E57.
Okay, so it's 144T.
I think it's just A, Julian.
Oh, sorry.
Yeah,
A?
the T is just the T is just saying that it mutated T, but originally I think you'll just be looking at the A.
A, right? Okay. So one.
Okay.
Okay.
So this
Give me a
one
second. It's
was
144, right? 50, 48.
So for this one, do you use the number that we wrote on my mutation, right?
Yes, we did.
Not like-
Because this one doesn't have the methionine. It starts on valine. So it's gonna be on exactly on that number.
Awesome, thank you.
Okay.
And then we would highlight that specific.
Just that one.
Okay.
Wait, should it be the same letter?
Oh shit.
No, it's not gonna be an A if your mutation was an A. It has to be what it was originally was.
Okay, I see.
It's the wild type one, right?
Yes, correct. It's the wild type. So say if yours was an L before on wild type and then it switched to alanine, you're looking for that L then.
It switched from an E to an A.
So Ty,
Or
you said 157? So yours was a
It
K
was-
before.
it was E57A. E57A.
E57?
Yeah.
Yeah.
You see the headers and they had numbers, and then it has 150. Like for example, on the screen it says 151. So I think if you point on them, you can see how she has it on the glutamine, whatever, and it has 58. So you see
Yeah.
numbers too.
This one says 58. I think it's this one, Ty. I think it's E58A. Because
Mm,
We do the
57
number.
is 57. 57.
Yeah,
We do the number
57
minus
is-
1, or
It's a K.
Yeah, so I think it's 58 then. Oops, sorry.
so we add the +1 back in for this.
No, you don't. I think I
Okay.
think we accidentally messed up for the naming on that one. So yeah, yours would be
'Cause-
E58A then. And this is your amino acid before it changed to an A.
58. Okay. So just highlight the E then?
Yeah, just highlight the E. So hold on, let me try to highlight mine. Where's mine?
This is a little hard.
Okay.
Just
Is everyone good? Everyone highlighted? Just your mutation.
Wait, so you went from favorites to what?
To favorites to sequence.
Sequence?
And then from sequence, we only clicked on chain A.
Oh, got it. Okay. So what do you have to highlight? You just click it or what?
Hold on. Is everyone on the highlight?
Yes.
Okay, so we go to actions,
Oh shoot. Okay.
actions, and then from here, we're gonna go to,
Sorry, wait, I'm still doing.
okay,
Okay, wait, so we go, I'm on chain A, and then what do we do from there?
you look for your mutation. So what was your position of your amino acid?
Let me check. Let me see.
Oh shoot.
It was K130A.
Right here then. 138 right here. Mhm.
Let me see.
So where my E is, where it's highlighted, yours is the K.
Oh.
Mhm.
Okay, so then what do I do from there?
You highlight that amino acid,
I just highlight the K?
just the K.
Okay.
Okay, ready?
Hold on. I think I don't know. Mine's weird because it kicks me out of the picture with the your background. You know how your hair it's all black.
You click on action.
Won't let me. Like it's all black. Like it won't. For some reason, won't let me.
Oh, black, what the?
Yeah, I don't know. The heck. I might have to just hit it again.
Yeah, you're gonna have to restart it all over again then.
Yeah. All right.
Okay, so you guys want to keep on going, or do you guys want to wait for Julian?
We can keep on going.
Okay, so from here we're gonna go on actions, and we're going to go to atoms and bonds, and we're gonna click on show. And so actions, atom and bond, show. Is everyone good?
Yeah, I got it.
Okay, and then we're gonna go back to action. We're gonna put a label, we're gonna put residue, and then we're gonna go to 1-letter code and specifier.
And do we click on it?
Hmm?
Do we click on it?
Click on it.
Yeah. Or no?
And then we're gonna go to, hold on.
Wait, can you repeat that one again one more time? I'm sorry.
So on which one? The
one you just did, right?
Okay, so we're gonna go to atoms and bond, show.
Okay.
And it'll show. And then we're gonna go back to actions. We're gonna go to label, and then
Okay.
go all the way to residue, and then the 1-letter code plus the specifier.
Thank you.
And then we're gonna click on focus. Oh, we don't have to focus that much. So hold on. I don't know if you guys can take a picture of that to show. No, that's your. Ooh, not meant to do that. There you go. Okay. And now this is where we're gonna find our interacting. So we're gonna go to select. We're gonna go to zone. And from here, we're gonna click 4.0. And then we're gonna click select on all bonds, atoms of any residue in that selection zone.
What is this called to like title that picture that we just took?
What is that called? That one's, the one that we just took is just your amino acid. So I think you can call it, selected amino acid or wild type amino acid on the inactive form of CRP.
Okay. And then this step you're doing right now, I'm sorry, can you just like cancel and reopen that?
Yeah, go ahead.
Like,
Yeah, I
how-
can. So select. We're gonna go to zone. And here we're gonna put, it should say 5.0. I put 4.0 because that's the max of what we're looking for distance. And then we're gonna click on select all bonds, atoms of any residue in selection zone. Thank you, Jasmine.
and then it gives a whole new picture, kind of like zoomed in.
And then it's going to highlight the interacting amino acids. And from there, we're gonna go to actions, atoms and bond, show.
Oh, shoot.
Sorry. There you go.
Hi. Do we take a picture?
Hold on. And then we go to label, residue, again, then the 1-letter code and specifier. And you can take a picture there. So I don't know if we can get like a really good picture. We can find.
And what should we call?
Wait, you went to label and then what? Or what?
Residue. We went to label, residue, and then the 1-letter code plus the specifier.
Okay.
I wish we can change the color on this. We can be a little bit more visible. But I guess
What
not.
should we call this one?
So,
This one is going to be not interacting, but this one you can put residues nearby. Because right now we're going to do the distance.
so wait, can you go back like one step real quick?
To where?
So I'm at the part where it's only highlighted the like that specific one.
Okay, so you're going to go to zone.
Okay.
Okay, so the top one's already clicked, right? So that one you're going to put 4.0 instead of the 5.0, correct?
Yes, that is already done.
Okay, and then on the bottom, you know how it says select all items and bonds within the selected zone area?
On select, and then you go to what?
Oh, hold on. I'll try to.
Oh, hit zone. Okay.
Yeah, and you press OK. And it's going to highlight everything for you, correct?
Hold up. Let me take that
It
out.
should highlight the atoms and bonds nearby.
It doesn't do it for me. What the heck? I just had it run away. So you went one more time. What was it? You went to actions, and then you went to label.
We went to actions first, and then we went to atoms and bond and show.
Okay.
And then we went to action again, label, residue, 1-letter code plus the specifier.
Okay.
And we should be good at that point.
No, it's not.
And then you have to do the select zone part at the end.
So the 4.0 and the 5.0, that's already in there. So
Did he
But-
click on both or did he click on just the top one?
on both.
You only click on the top one. See?
Oh,
Mhm.
okay.
And you click on the select all bonds of any residue. And the, oops. Then now we need to do that. There.
Can I share my screen? Because mine's like, I don't know.
Yeah.
Yeah, because
Right.
let me simpler. Let's see. So it gives me this.
Yeah, and then press OK.
And then, oh, what the heck? All right. And then from here,
We still see the bond. Are you sharing just that or?
oh, I don't know how to, like, it's hard to maneuver.
When you share, try clicking on your desktop, like your whole desktop, not just.
Oh, I can't see what you mean. So let me share again.
We're just looking at that one part of the selection zone.
Okay.
Try again.
So, okay.
Okay. And then from there, you're gonna go to action, atoms and bond, and then you're gonna put on show. And then you're gonna go to action again. And then go to label. And then residue. And then the 1-letter code plus the specifier.
This one?
And there you go.
Okay. Okay.
Okay.
Let me stop sharing.
Let me share mine again.
Thank you.
Welcome.
Make sure you take a picture of that, Julian.
Yeah.
Okay.
Okay.
Like zoom in and
From
everything.
here, we're gonna actually stop highlighting everything. So clear selection. So go to select, clear selection. So depending on your residue, you're gonna try to find ones that are interacting. So mine, the
Should we
R,
remove the ribbon?
huh?
Should we remove the ribbon?
If you want to remove the ribbons of chain A and chain B, you guys can. So go to chain B, ribbon, and then hide. And then go to select, chain B.
You can just go to action, ribbon, hide.
For all of it?
Yeah, it'll take it all away.
Oh, okay. And then just go to select and then clear. Ooh. Where's there? There you go. So you're gonna try to find what your amino acid is interacting with. So mine would be R82B from the chain B, and I think I would have to do the K130A and then maybe the Q. But we have to measure those distances, so we're going to
So you're looking for molecules that are separated?
not separated, but you know where your R group from your amino acids at? You're gonna try to find other R groups of other amino acids that look close to it. So like this one right here, and this one, maybe this one, and then for me, I would think it's this one too.
So if they just look physically close, that's enough.
Yeah, but we're going to actually find the distance to see if they are.
Okay, because I see a couple that are looks like they're floating on their own. They're not connected to like the main big chain.
Yeah, because there's two chains. There's chain A and then chain B.
Okay.
So
So the ones that you're looking at that are nearby yours should be what color?
it can be anything as long as it's, I think, the R group of the amino acids. So mine is oxygen, but these are like hydrogens, but they're still interacting. You get me?
How do you know it's the R group?
Because this is for mine, which is glutamine. That's
Yeah, I think you have to go look at Google. Go to Google and look what your amino acid is and what the R chain is, because there's the backbone. The backbone is, I think, made up of the amine and the COOH group.
I have a question real quick before we
Yeah.
continue. So how do you remove the like the rest of it, except that part? Because I'm trying to click on everything, I can't find it. You put select,
Sorry. Oh, I don't know what I selected. What are we trying to do?
like remove like all the background except
Okay,
that
so
part.
Ribbon.
yeah. So Christian said is go to actions, ribbon, and then hide.
Okay, actions, ribbon hide. Actions, ribbon. Hide. Got it. Okay, thank you.
Okay, and then we're gonna start finding distance. So for distance, you don't need all of your R group. You only need the atom here and the atom of your R group. So the way for Mac, the way we do it is control and then click on one of the atoms and then control shift and then click on the other atom.
Do you mind if I share mine so I'm kind of confused on which one it would interact with?
Yeah, go ahead.
Okay. So this is mine.
Which one? So
And that's this one right here.
the R?
Yeah. I don't know how I would.
Oh, it actually tells you it's an R.
Yeah. When you put the label with that says the one letter and then the residue, it tells you.
Oh, okay. I see my.
Okay, so your R, and I think that blue is your R chain. So I think it's interacting with
this one here.
Yeah. So it's interacting. Hold on, let me annotate it right now. Okay, so yeah, so this is your backbone, right? And so this is your R group. So I think it's interacting with this here. And then we think, what's this? Hold on. Maybe, wait, what R group is this one right here? This one right
Oh, I can't see your mouse. Oh, this one?
Yeah, what R group is that one? No, that's a Y. That one, no, that one's that's the R group. So this is lysine. Okay, maybe it's interacting. Can you move it around so we can see? Okay.
It's just gonna move.
I know. Yeah. Hold on, let me try to delete what it so it doesn't make it confusing. So
it's still this here.
This one for sure. It's that red one that I told you.
This one?
Yeah, that one I meant, sorry.
So Lizette has arginine. Does she have to check all the all the nitrogens on her R chains?
No, you're just trying to see ones that are close to the R chain.
Because you know how you look at
this
the
one too,
they have 3 nitrogens. So does she just have to check one nitrogen, or does she have to check all 3 nitrogens?
She doesn't. No, you just check which ones are closer. So
okay.
Yeah. So is it leucine? Leucine? I think that's leucine. Maybe leucine is interacting this white one with yours.
These two right here?
Yeah. Oh yeah, that looks really. I think this is interacting right there.
How is it that you do the tools?
Oh, I'll do that right now.
Do you just have to, so do you have to check with the distance?
Yeah.
That's you have to check with the distance.
Yeah.
Okay,
so,
here. Let me sharing.
So let me go back to mine. Okay, so for Mac, it's control, click, and then click on, well, for me, it would be this one, and then control, shift, and then click on that atom. Yeah, you only want to select the atoms. You don't want to select the whole thing.
You said for Mac it was Control Shift Click.
Click and then Control Shift, not Control Shift, Control Click on the other one.
PC, it's the same. Control click and then Control Shift click.
Okay. Thank you, Christian. And from here we go to
Do you mind if I share mine so you can tell me if I'm doing this right or
yeah, go ahead.
share screen? And then, okay, so mine this one, right? The 144. And it will be interactive with this one, right?
Yeah. I believe so, yeah.
So like this one.
Mhm.
Well, that's like the only one there is. Or because this is it, right?
Yeah. Maybe that, wait, hold on. Maybe this one right here. The what that is that the L140? What is it?
Which one you talking about? Oh
There's
shoot.
hold on. What is that? An L? Hold on. Can
Sorry.
you zoom in?
I guess I can't. Okay. All right. I'm gonna press.
Oh, there you go. Sorry, I'm not trying
Sorry.
to be rude
That's
or
okay.
anything.
So
Yeah, maybe the Lucy
pizza, right?
that one. Yeah, and maybe that L148.
It's on 8.
Maybe that one. Yeah.
Oh, it's pretty far.
The top one.
Oh shit.
Maybe? Yeah, the top one right here. No, not this one. So
Oh, I
back
can't.
to the L148. I
Okay.
think it's probably the top one that interacts with it. But we're gonna see right now what the distance. Yeah.
Okay.
So
Let me.
we're gonna do distance now, so you guys can make sure it does. Hold on. Okay. So
Are we just looking for pretty much the one closest interaction we can?
what are we looking? Wait, can you repeat that question?
Are we just looking for the 2 points that are like the closest together on that R? We're not looking for multiple different.
Yeah, you're just gonna try to look for which ones are the closest. Yeah. So mine are highlighted. Like I said, it was Control click on one and then Control Shift and then click on the other one.
Okay.
We go to Tools and then we go to Structure Analysis. And then we go to Distance. And then from our distance, it's all good. We don't, oh, hold on, I'm trying to move. Put this down here. Okay, so from here, we're just going to create. And it's gonna tell us our distance. So he said 2.5, but that's really close together. So we know that one's interacting. So it's gonna be 1.740 Armstrong. So that one is for sure is interacting.
What's the threshold for interaction?
The threshold he said was 2.5 to 4.0.
Okay,
But
so
he said
mine's
anything-
1.6.
Yeah, he said if anything's like really close, like below 2, it's most likely interacting.
Do we need a picture of this?
Yes, we do. So hold on.
Do you guys know how to move, like how to move your screen like from one side to? I get stuck like rotating it. I'm trying to like move to the side.
I use my mouse and I just hold down and then I rotate it.
Oh, yeah, my mouse. So, quick question. So once you click on the first one, and then if I click to the other end, I hit Control Shift. You said
Control Shift click on one, and then Control click on the other.
Okay, let's see if this works.
And that's called what? Residue interaction?
That's interacting residues. Yep.
Got it. And then you'll go favorites to look at distance.
Oh no, you go to Tools and you go to Structure Analysis, and then you go to Distance, and then from here it says Structure Measurement, and you just press on Create, and it creates your distance. It'll give you even like the actual measurement.
Okay,
So
I'm
mine is 3.34.
That's good. That's so good.
Okay.
Someone was gonna say something.
Yeah, I'm confused on how you found that. Can I share my screen?
Yeah, go ahead.
Okay. So I have the K130A, right? So,
So what's your R group? Is it that blue on top? Think that one is.
Where?
That blue, the nitrogens? Is it? Yeah. Wait, hold on. Let me look it up online. I'm pretty sure K is lysine, and I think that one is.
The threshold that he said again that it had to be for interacting.
2.5 to 4.0 Armstrong.
Okay, thank you.
So this is depicting.
Oh, hold on. Wait, try.
This whole like image is depicting our amino acid and the residues, right?
Correct. Of the inactive form of CR. Okay.
Okay.
So Jasmine, that nitrogen, the one that I told you that are on top, those are your R groups. So it's interacting with those oxygens of what amino acid is that? So control click on that on the hydrogen.
Where my mouse is at right now?
Yeah. So control click on that and then control shift and then click on that red oxygen right next to it. No, not that one. The one
over here.
Yeah.
Control shift.
Control shift, click on it.
Okay. Oh, wait.
Oh. That one, control click. And then control shift, click on that one.
It's not letting me.
It's not?
Control shift, right?
And then click on it while pressing control shift.
Okay, let me try it again.
And
I don't. It doesn't.
there you
Okay,
go.
I got it.
And then go to tools. Go to structure analysis. And then go to distances. And then it should give you like a little tab. You probably can see it, and then you just press create.
Okay.
And it's gonna give you like the distance. Yeah, that's perfect. Yeah, I'm just interacting. What is that? I think it's glutamine.
And lysine?
And lysine, yeah. Just make sure you write down what position the glutamine is.
So I'm gonna need this, right? So should I take a picture of this?
Take a picture of that, yeah. Okay, what? Julian, you had a question?
Hold up.
Oh my gosh, what just
Oh no. Is it so when you find one, that's it, or we keep clicking on the like around them to see if there's more than
You
one?
have to try random ones. If you feel like they're close enough, try finding the distances for them.
So I'm only asking that because I don't know if looking for a specific one, you know?
Yeah, you're not gonna look for a specific one. You're just gonna look for ones that you feel are like somewhat close. So I feel like this lysine group is pretty close. So I'm gonna do the same for this one. So
Here, Dad, eat these first.
click,
Dad.
control shift, click on this one.
And
Go to tools, structure analysis. Create that one. That one's a 3.390. That one would be considered interacting.
Okay, so there's not a limited amount we need, or
There is not a limited amount. It's whatever you think is close to your R group. And then you measure it.
And the R group is always like the blue area?
Not necessarily. Sometimes it can just be carbon. You just need to look at your amino acid. If you don't know what the R group it is, then I recommend looking online.
Yeah, I'm lost on that part. I thought it was looking at the one that's labeled with the letter R before it.
The letter R?
like on your screen, see the tan labels? Like in 133A and some of the, yeah, I have one that's R and then a number. I thought that was my R group.
No, that's actually like this. That's an amino acid. So it's this is
Okay.
just, yeah.
Then I measured the wrong bit. I'm not sure where my R group is then.
Okay, so what's your one letter residue?
Where do I check that?
What was your mutant
My
before it
mutant
changed to
is
alanine?
E77.
Okay, so yours is glutamine, I believe. Hold on. Just making sure. No, yours is glutamic acid. So glutamic acid. Then you're gonna have to look at the structure then for that one. So let's see, glutamic acid. Structure. So I believe, yeah, your R group, it's gonna be 2 oxygens and a hydrogen. So you want to screen share so I can help you look for your R group?
Yeah. Screen share.
Hold on. Am I screen sharing still? No. Okay.
No, you're good.
Okay.
Can you see it?
Yeah. So let's look for your residue. So it was what's it?
E77.
E77. I don't know if it's that one right there. E77. There's an E78. Let's see. Hold on, I'm gonna try to look for it highlighted. So go to favorites.
Here, let me just double check this really quick.
Okay.
Here, give me. I'm gonna share my Word to make sure I have the right mutant.
Okay.
Because I very well could be off one. Okay. So there's my mutant right there.
Okay, so 80, 79, that's 78. 78, 77.
80.
Yeah.
So I was right?
Mm-hmm. 80.
Okay.
90, yeah. Okay.
Oh, sorry. Do you need to see that again?
No, I was just making sure.
So it is E77A, right?
Yeah, hold on. It's because there's a lot of atoms. Can you screen share so I can, hold on. So go to favorites. Go to sequence. Click on chain A only. And then look for your amino acid and then just highlight it. There you go. Okay, can you go to, try to zoom in? Oh, there. Oh, okay. Just try tilting it around. Okay,
Is
so
it
that
right
red,
there?
yeah, that red is gonna be your,
that?
Yeah.
Or is it this?
It can also be that too.
Okay, so I'll need to check both distances.
Yeah.
And then what's the threshold range to use them?
2.5 to 4.0 Armstrong. If it's lower than that, then it's still interacting.
Okay. All right, cool.
Hey, Marilyn.
Yeah.
I think I did it. You want to check it real quick?
Yeah.
Thank you. Can you see that?
Yeah.
So this is what I have, these two.
Yeah, that looks good.
Yeah? Cool. Thank you.
Then for his, like the one that's 1 point, I think it's 6, That one wouldn't
Yeah.
be interacting, right?
No, that one is interacting because it's really close. He said 2.0, 2.5. I don't know why, but if it's anything lower than that, then it is interacting.
And what if it's like the 3.6 one?
That one still is interacting because it's lower than the 4.0.
It just has to be lower than 4.0.
Yeah.
Okay.
So does it have to be red, like the little tip?
No, it just depends
Because
on
mine
what
are
your
just
art group
white, and
is.
I'm like, I don't know if I'm.
No, it just depends on what your art group is.
I just did it like exactly. Like, I just saw her color, so I just did like the same thing. I just saw like the same stuff around there. So I just tested it. That's all.
Okay.
Okay, bye, Julie. Just do the same for this, for the inactive, for the active.
Can I share my screen real quick just to double check the R group part?
Yeah, go ahead. So what's your one letter?
Is K. So
Okay,
lysine.
so it's lysine. So lysine has nitrogen, so it should be blue.
Yep. Let's see. Oh shit. So
Is
that's
that
what
one?
I have. So there's
Yeah, that's good.
So the R group would just be this blue
Yeah.
thing? Okay, just making sure. Thank you.
Welcome.
Can you check mine now?
Yeah.
Okay. Let me share.
So what's your one letter?
Q.
Q. So Q is Q is glutamine. Okay, so glutamine. So glutamine should have a red and blue. It has oxygen and so yeah. So I think it's that one on top, maybe.
Oh, the top one.
Mm-hmm.
Okay. Okay. Thank you.
You're welcome. Yeah, if you guys ever feel unsure what your R group is, you can always just look up what the letter code stands for and then just look for the structure for them.
Okay, can I show mine one more time? Sorry,
Yeah,
I just
go ahead.
because I don't know. I think I'm doing this wrong. I don't know. Okay, so see, I don't know. Like you guys is like everyone else has like a red little cap thing. So I don't know because mine's A144, right?
Yeah, you
But I
have
don't know if-
alanine. Alanine has carbons.
Okay,
It doesn't
so
have
it's not
an oxygen. No, you're good. Yeah, you're good.
okay. Okay, I'm like
Yeah.
I don't know because I keep seeing everyone like has like a red one. I'm like I don't know. Okay.
Yeah,
Thank
you're
you.
good. You're welcome. That's it. I think. Oh, let's see. Let me check the distance for this one to this one.
So are we supposed to find like interacting parties in the active? Didn't he say there's not supposed to be any interacting
And
parties?
inactive? No, there is supposed to be.
Yeah.
This one's the inactive. This one is interacting. That's what keeps it stable. So that's why it's not functioning.
No, I mean in the active,
In the active, yeah, in the active, this R of the B chain, I think the one that's interacting the most is the one from the B chain. That one shouldn't be there.
Okay.
Yeah.
So what will we just say there's nothing interacting?
I would make sure by finding distances just in case.
Okay.
So how many interactions should there be? Like 5 or more, or just a couple?
It's as many as you find. So I only found 2. You guys see how I only found 2? Because this last one, it's too far. It's over the 4.0.
How do you find distance again? You hit tools and then
Go to tools, and once you have them highlighted, only the atoms. So
yes.
control click on that one and then control shift click on that one.
Mhm.
You go to structure analysis and then you go to distance and then
Oh, structure analysis. Oh,
Yeah.
okay, got it. Okay, got it. Thank you.
okay, so let me take a picture of this. I need this. There you go. Okay, and that's all you guys need to. I don't know if you guys want to do the active one right now, or if you guys are like, I already have my video, I can do it by myself.
I think we can probably figure it out.
I agree.
And
Yeah,
if you guys have any questions, I'm always down to help you guys.
Did you were we gonna do another Zoom one? Because I have most of this stuff done, like the sequence analysis and then the protein analysis. I just needed to find the distance, but I'm having trouble on the activity data on the last part with the DNA binding one.
Mhm.
I was just having trouble on how to interpret that one. I already did all the colors, like the blue, white, and small and the large. It's just the last part and the interpretation part.
Yeah, we can do another session if you guys want to do like next Sunday.
We should do a poll, like,
Or
you
I
can
can-
do a poll here
Or I
if
can
you
do-
want.
Mhm.
Sorry, go ahead. That way, if we can get together before the weekend, like if any time before the weekend works.
Yeah.
No, yeah, I agree.
Mhm.
Because we want to try to get it done like as early as possible, you know?
Yeah, just because it's due on Tuesday.
Yeah. What is the activity data for? What is that for?
Oh, sorry. The activity data is for the paper, and it's supposed to be on the poster too.
But I
don't-
think we can also include that in our PowerPoint.
Okay, so that can be on our PowerPoint.
Yeah, I mean, I would want to add it because 15 slides.
Yeah, it's a lot.
Yeah,
Yeah,
that's true.
so I think if we can add as much.
Yeah, because
Yeah.
I